Hello everybody, welcome to the video about carbohydrates, the first of 4 macromolecules that we'll be talking about individually. By the end of this video, we should understand the structure and function of carbohydrates. Carbohydrates are hydrophilic organic molecules. If you recall from a few videos back, hydrophilic means they're able to dissolve in water, and organic means they have a carbon backbone. Examples that I'm sure everybody is very familiar with are sugars, things that taste sweet but aren't artificial sweeteners. That's a bit different. So table sugar or maple syrup or sweet peach or strawberries. And also starches like potatoes or wheat. If we're trying to figure out if something is a carbohydrate by the chemical formula, look for this ratio of molecules or of atoms. We're looking for carbons, hydrogens, and oxygens. We won't see any other elements in there. We won't see any nitrogen, we won't see any sulfur, we won't see any phosphorus. Okay, so just carbons, hydrogens, and oxygens. And we'll have twice as many hydrogens as oxygens and about twice as many hydrogens as carbons. So glucose is one that we talk about a lot, and it has 6 carbons, 12 hydrogens, and 6 oxygens. So you see how that rule applies. We have double the amount of hydrogens as the other two. A lot of times, carbohydrates end with "-ose at the end of their name. Some of them have sacra as the prefix, but not all. "-ose is a really good rule of thumb, though. The 3 most important monosaccharides that we're going to talk about are glucose, galactose, and fructose. But first of all, let's talk about this word monosaccharide. Remember that mono means one, and saccharide is sugar, so one sugar. So this is the monomer, and then the polymer is going to be the full carbohydrate. Okay, so monosaccharide, one sugar. The 3 monosaccharides that we're interested in, glucose, galactose, and fructose, all of them have the same chemical formula, C6H12O6. So remember when we looked at the isomers before? These are isomers of each other. They have the same chemical formula, the same elements, the same atoms, and the same ratios, but they're arranged differently. So let's look at what the molecules look like. Each one of these, if we recall, has 6 carbons. This is a place where some people get really confused. I know when I started studying chemistry and biochemistry, it took me a little while to catch on to this. When I first glance at this monomer, this glucose right here, I only see 1 carbon, and it doesn't make sense to me that the formula is C6H12O6. What you need to remember is each one of these corners is also a carbon, and it's unwritten because it's hard to write a letter on a corner. So we have 1 carbon, 2, 3, 4, 5, and 6. And this one we call glucose. Here we have galactose, and it's the same way. It has 1, 2, 3, 4, 5, 6. So both glucose and galactose are 6-sided rings. 1 side, 2 sides, 3 sides, 4 sides, 5 sides, 6 sides. Pretty obvious, right? So what's the difference between the glucose and the galactose? Well, we have the same functional group up here at the top. Same oxygen at this corner. Hydrogen and oxygen, hydrogen and oxygen, hydroxyl group. Hydroxyl group and hydrogen. Here's where it's different. Here in galactose, the hydroxyl group is up and the hydrogen is down. Here, the hydrogen is up and the hydroxyl group is down. That probably seems like a really small difference. In reality, it changes how the monomers can bind together. And so it really does change the chemistry of these 2 monomers. Fructose is a little bit different. Fructose is a 5-sided ring, 1, 2, 3, 4, 5. I've got my first carbon projecting up here, second carbon, third, fourth, fifth, and then the sixth one projects down here. We could also count up and prove to ourselves that they have the same number of oxygens in all three and the same number of hydrogens in all three. You are welcome to do that on your own time. I won't do that here in the video today, though. Disaccharides are formed from two monomers. So sucrose, everybody is super familiar with. This is plain old white table sugar. Maybe you added it to your coffee this morning. And we're going to add together a glucose and a fructose. And when we put them together with this bond, remember this was a dehydration synthesis reaction that put these together, we have the disaccharide of sucrose. Let's break down the word disaccharide. Remember we talked about mono, one saccharide, sugar a minute ago. This is a disaccharide. So di is two sugars, disaccharide. So sucrose, table sugar, glucose, and fructose. Lactose. Lactose is interesting. Lactose starts with a glucose, and then it has a galactose. Notice that the bond in this one is different. If a person is lactose intolerant, they don't have an enzyme that can break this specific bond. It looks really similar to maltose down here, which is a glucose plus a glucose, except the bond is upside down. And that results from galactose having the switch of that hydroxyl group right here. Okay, so this plays a big role in what enzyme is needed to break these apart and how easy they can be digested, and even if we're able to digest them at all. If you're lactose intolerant, being unable to break this bond is going to make you feel really crappy if you eat ice cream or some cheese or regular milk. All right, so monosaccharide, single ring structure. Disaccharide, two rings. We just looked at that. And oligosaccharide, oligo means a few, is three or more monosaccharides. Some people define it as at least 10 monosaccharides. A little bit of disagreement in definition on that one between sources. And then when we have big molecules, they're polysaccharides or many sugars. So generally they have at least 10 of those ring structures to make them up. So glycogen, you may not have heard of before. Glycogen is really, really important. Glycogen is how your body stores carbohydrates. There are two places that glycogen is primarily stored. One is your liver and the other is your muscle. So your liver and muscle tissues store this glycogen, and when your body needs energy to do something, maybe to go for a walk, for instance, your body is going to first break down the glycogen that's stored in your muscles and stored in your liver. And as that energy is released, you are able to do the things you choose to do. Let's imagine for a moment that you are running a marathon. Have you ever heard of a runner say they hit the wall? When a runner says they hit the wall and they suddenly feel very fatigued, what happens is they've used up all their glycogen. They don't have any more stored carbohydrate in their body that they're able to convert into quick energy. And at that time, metabolism does some changes. It's really cool biochemistry stuff. But then that's when your body starts burning fat instead of carbohydrate as a fuel source. So starch, I think we're pretty familiar with in some of the foods that we eat. Potatoes and wheat, like pasta and rice, are things that have tons of starch. Very easy for us to digest. Cellulose, we can't digest, but other animals can. So cows, sheep, horses, things like that, they're able to digest cellulose. Even that is a little bit of a misnomer. The animals that can digest cellulose, they aren't actually the ones digesting it. They have bacteria in their stomach and in their gut that do the digesting. The animal itself doesn't. It's the bacteria that they're hosting. So if we go back to glycogen, remember glycogen is the carbohydrate that gets stored in muscles. One of the features of glycogen is it's a branched chain structure. So we have a chain going on here and then this one branch coming off to the side. And magnified here, we can see chains and branch, chains and branch. So the whole idea of carbohydrates is we can get quick energy from them. Our bodies usually are very, very efficient at burning carbohydrates to get energy. The way it works is through a three-step thing. I hope you guys remember this from high school biology or an earlier biology class. First, we undergo glycolysis, and then the Krebs cycle, and then the electron transport chain. And by doing that, one monosaccharide is broken down into ATP, and that's what we can use for energy. It's important to note that no matter what monosaccharide we're talking about, whether it's galactose or fructose, all of them are converted to glucose before they go into glycolysis. We also have something called conjugated carbohydrates. Conjugated carbohydrates are where carbohydrates are put together in a complex with either a lipid or a protein in order to do a specialized job. So glycolipids are a combination of a carbohydrate plus a lipid, and they're attached to our cell surfaces. They stick out from the cell membrane and they're used in some signaling and cell recognition. Glycoproteins and proteoglycans are very similar. They're both a mix of protein and carbohydrate. Glycoproteins have more protein. Proteoglycans have more carbohydrate. The glycoproteins are also on cell surfaces, and they're very important in the mucus of the respiratory tract and the digestive tract. So this is really gross, but glycoproteins are pretty much your snot. If you sneeze and a bunch of mucus comes out, those are glycoproteins. They're very sticky, and they help to catch pathogens and dust and things like that that you may be breathing in when they're in the respiratory tract. Proteoglycans are more structural. They hold cells together. They're also the thing that fills up your eyeball, and they're also the thing that you find in your joints to keep them lubricated. You might know somebody who gets fluid injected into their knees, for instance, because the fluid in their knee has degraded with age. What they're replacing is proteoglycans. So that's a quick summary of carbohydrates, and we'll be coming back with the next macromolecule in the next video.